Dear colleagues and dear students, my talk today will be about the genetic and proteomic changes in cancer, and the contents of my word will be about what is cancer and other topics. I will say it on the spot. So what is cancer? The tumor is a mass composed of different cells that differ from the progenitor cell. We have a colony of the cells over here, and it differs from benign tumors that it can invade other tissues. Most of the tumors that we find are not malignant tumors, but they are benign tumors. The only difference is that malignant tumor can invade other tissues, while benign tumors do not. There are more than 100 types of cancer. We have cancers that arise from the epithelial tissue, which is called carcinoma. We have adenocarcinoma, basal cell carcinoma, and squamous cell carcinomas. And other cancers that originate from the connective tissues, and they are known as sarcomas. We have lymphomas from the lymph nodes and lymphocytes, and we have leukemias from the blood cells. We have multiple myelomas, melanoma, brain tumors, germ cell tumors, neuroendocrine tumors, and carcinoid tumors, which grow very slowly. The other topic will be etiological factors for the cancer. So what are the etiological factors? We have three main groups for the etiological factors. The three main groups are the radiations, like the UV radiations A and B, the chemicals, and the viruses. And I would like to say there isn't a certain compound that causes cancer by itself. There must be many compounds which act together to form the cancer. So they act in what is called double hit hypothesis or multi-steps. One of the chemical causes initiation, and this initiation is a sort of mutation in the cells, in the DNA of the cell. We can live like that without any symptoms of the disease. If this mutated cell is subjected to another carcinogenic compound like chemicals or radiations or whatever, another mutation will take place, and this is called promotion. Still, the cells are limited. The growth of the cell is limited. Although it is more than the proliferation is in a more rapid way than the normal cell, but still the tumor didn't appear. After that, we are subjected to other chemical or UV light or radiation or virus, and this causes a sort of progression for the cells, and so in this stage the cell will proliferate in a very fast way, and they will form the malignant tissue that we see. And this is a sort of diagram to show these steps of initiation, promotion, and progression till the tumor invades through the lymphatics or through the blood cells. These steps causes mutation, increasing capacity of proliferation, survival of the cells, invasion of the cells, and metastasis. And so this is called the tumor clonality, which is the clone of the tumor that differs from. The other topic will be about the properties of cancer cells. So what let a cancer cell survive and proliferate? They have certain properties like density. They don't have density dependent inhibition. Our cells grow in a certain way that they know that they are in a crowded area, so they grow in only one film of cell in the culture. But the cancer cell, if I culture a cancer cell, they don't have the density inhibition property. They don't have the property of knowing that they are in a crowded media, so they grow overlapping each other, and this forms the tumor. Also, we don't need to put growth factors in the culture because the cancer cell has its own autocrine growth factors, like the RAS gene, like other stimulation growth factors, and inhibition of tumor suppressor gene, and so on. Also, they have reduced expression of cell adhesion. They are not adhesive to each other, but they are loose so that they can immigrate and flee away from the tumor itself. Not only that, they have excessive protease digesting extracellular matrix. So they can digest the proteases outside the tissue that adhere the cells to each other, and they have a reduced ecadherine, which is the gum that adheres the cells with each other. So they are loose. They can migrate and invade other tissues. Not only that, again, they have the property of invading or forming new blood vessels to invade through the tissue and to get its nutrients and the oxygen they need. So they form very minute blood vessels, which is a property called angiogenesis. Also, they fail to form normal differentiation. We have the stem cell, as Dr. Nagwa said. The stem cell is differentiated to different cells in the body. So during this stage of undifferentiation, the cell proliferates in a very fast manner, but when it reaches the proliferation, the growth is slower. But a cancer cell does not differentiate, and it goes over through proliferation in a very fast manner. Also, it has the property of failure of undergoing apoptosis or survival. So if a cancer cell is mutated, it survives as it is and does not apoptose, and it does not die. That's why the tumor arises. So the cancer cell can undergo any obstacle it meets. The fourth topic will be about heredity and sporadic cancer. What is the difference between hereditary and sporadic cancer? And I prefer saying familiar cancer and not hereditary cancer, because cancer might be found in certain families more than the other. And this represents only from 5 to 10% of the total cancers that we meet in our life, but most of the cancers are sporadic. What is the difference between a familiar or hereditary cancer and a sporadic cancer? As I said, we have to take the first hit, which is the initiation of the cell by a sort of mutation in the cell. I might inherit this mutation from my parents, but this does not mean that I must have the cancer. But it is the first initiated process in cancer formation. On the other hand, the sporadic cases, they don't inherit this type of cancer. So if I make a sort of genetic screening in the sporadic cancer, we find that the mutation will be found only in, I don't know if it is clear here or not, the mutation will be found only in the tissue that is affected with cancer. But if I make a screening for the genetic mutation in an hereditary cancer, I found it in all the cells of the body. This is the main difference between both types of cancer. We have in the hereditary cancer, the first mutation, as I said, it is inherited from the parents. Other relatives might have the same mutation. The cancer occurs at an earlier stage than the sporadic cancer because he has already inherited the initiation. Sometimes it is more aggressive than the sporadic cancer. It may respond to different treatments than the sporadic cancer. And individuals who have this inherited gene might have different types of cancer. As regard to the sporadic cancer, as I said, 85% or even less of the cancers are sporadic. And sporadic cancers are believed to be a cause of genetic damage caused by exposures, dietary factors, hormones, normal aging, and other influences. Again, this scheme shows the multi-step models for developing a colorectal cancer. The colorectal cancer in most people starts with a mutation in the APC gene, and this mutation is not cancerous, but it forms a sort of a polyp. This polyp is a benign tumor. If it is subjected to other mutations, it will form adenoma, which is still a sort of benign tumor. Further mutations like the RET gene or other tumor suppressor genes like the DCC will form a larger tumor, which will be after that a malignant tumor if I'm subjected to further mutations like the P53 and additional mutations. So this scheme shows me that a single tumor single mutation, can never cause malignant tumors. What are the genetic changes in cancer? As I said, there must be many mutations, and these mutations are attacking the DNA of our bodies, and they code different polypeptides in our body. And of course, I will not talk about the gene, because most of you know what is the gene, but I will talk about the genes that are related to cancer formations and what is meant by gene expression. I want to say that gene expression is affected by many factors, and gene expression means that the DNA is, after being replicated, it is transcribed into RNA and then translated into protein. I care for this protein, which is the functional unit. I have many genes that are related to cancer formation, and I will say a very quick hint about the mutation. I have, as I said, the chemical mutation, the irradiation, and the spontaneous mutation, and every type of tumor has a different number of mutations different than the other. As we see in this figure, we found that a certain tumor requires 11 mutation, another one requires 6 mutation, and another requires 2 mutation. And mostly the lymphomas and the leukemias require very minute number of mutations. That's why they are present in infants more than grown-up people. However, I want to say that the cancer is a disease of grown-up people, but solid tumors attack the grown-up people, not the liquid tumors, because the solid tumors require many types of mutation. So I'm subjected to many types of mutation. Whenever I grow up, I'm subjected to more types of mutations than the infants. Again, the types of mutation are either a duplication of the chromosome itself, deletion of certain types of certain part of the chromosome, or inversion of the chromosome, or translocation of the chromosome. What are the molecular nature of cancer? We have 3 categories of genes that cause or protect us from cancer. The first one is the oncogene, which is normally present in our body as a proto-oncogene. It is very important for our body. But once it is subjected to a certain mutation, it is converted to an oncogene, and it starts to damage the cell. It's like a thief that attacked the cell. This thief needs a policeman to protect us from his effect. This policeman is the tumor suppressor genes, and we have some children inheriting certain mutations in the tumor suppressor genes. That's why some infants get cancer very young, at a very young age. The other type of, or the other category of genes is the mismatch repair genes, and these are involved in the checkpoints in the cell cycle. The role of this mismatch repair gene is to recognize that a certain mutation had happened to the cell, and I have to stop the cycle, the cell cycle at this part, and to repair this mutation. And if this mutation is not repaired, the tumor suppressor gene takes the cell to the death or to what is known as apoptosis. These are the checkpoints that are present in the cell cycle. We have the gap 0 and the interphase gap 1, G1, and the G2 checkpoints and the spindle assembly checkpoint. And each tumor suppressor gene and mismatch repair gene are responsible for repairing the mutations of the cell in these checkpoints. And again, if these mutations are not repaired, the cell will suicide or die. So what is the action of the tumor suppressor gene? Sorry, of the oncogenes. As I said, the oncogenes are normally present in our bodies in the form of proto-oncogenes, but they can be mutated by viral infection or other chemicals or whatever, or chromosome translocation or whatever from the effects. Let us look for this scheme. We found that this is the cellular membrane, and I have the growth factor receptors on the cellular membrane. They are formed of three domains: the intracellular domain, the transmembranal domain, and the extracellular domain, and they are always in dimers. When the growth factors come and settle on this receptor, the receptor become attached to each other, and they are activated by thymidine kinase to be phosphorylated and to form a cascade of reactions and giving a signal for the cell to divide. This is the normal condition, but there are some abnormal conditions when the cell, the growth factor receptor does not have the extracellular domain, and it's not in need to add a growth factor, as I said before. It is activated and it starts giving a cascade of reaction till it reaches the nucleus to tell the cell to divide. Another type of mutation is that these are always, they are neglected. They don't need to have growth factors, and the cascade of reaction continues till it reaches the nucleus and give an order to the cell to divide. So these are the mutated forms, and these are the natural and healthy forms. This is the action of the oncogene. On the other hand, what is the role of the tumor suppressor gene? As I said, the tumor suppressor gene is like the policeman that protects us from the effect of the thief. They control the cell cycle at the checkpoints to ralenti or to slower down the cell cycle at the checkpoints to discover where is the mutated part of the DNA and to repair it. We have the retinoblastoma gene, which blocks the cell at the G1, and the P53 gene, which retains the cell progression, and it has another effect, is to take the unrepaired cells to the apoptosis and to die. The third category is the mismatch repair gene, and the mismatch repair gene knows where is the mutation. How can it differentiate the mutated part? It differentiates the mutated part because we have the template DNA. When it is formed, it is methylated at certain parts, and this template DNA must not be attacked by the mismatch repair gene. It attacks the newly formed DNA strand, and it cuts the mutated part, and then the polymerase enzyme comes and put the correct nucleotide in the part that has been cut, and the ligase enzyme attach the DNA again, and this is the way of repairing a mutation in the DNA. What is the application of genetic changes in cancer? Of course, knowing all this knowledge, we have to know how can we use all this knowledge in a very bad disease like cancer. How can we protect ourselves? How can we prevent cancer? How can we detect it at early stages? How can we make genetic diagnosis? And how can we make targeted therapy or personalized therapy or tailored therapy for the cancer based on the genetic changes? Let us take the first thing, which is prevention and early detection of cancer. We have some hereditary genes that are transformed from a mother or from a father or from either of the parents to the offspring or the newly born child. This will be with him all his life or with her all his life. Let us say the hereditary and breast the hereditary breast and the ovarian cancer. We have the BRCA1 and the BRCA2 genes. They are mutated in some families. But again, I want to say that this mutation does not mean that they must have cancer at a certain time of their life, but they have took the first initiated mutation in their life, so they are subjected more than others to develop cancer. We have also the RAD51C and the RAD51D, the BRIP1. They are linked to ovarian cancer. We have the Lynch syndrome, and the Lynch syndrome is a sort of mutation in the mismatch repair genes, and they subject people to many types of cancer like colon cancer, ovarian, pancreatic, gastric, small intestine, central nervous system, and renal and subcutaneous cancer. We have the Codon syndrome and the from any syndrome and others. We have the myeloid neoplasm, which has certain mutations like the GEBPA, and we have the myeloid neoplasm having other sorts of mutations in other genes, the myeloid neoplasm in other organs, and that's why, as I told you, they are the tumors of infants. Also, we have other mutations in the mismatch repair genes. We have, as you can see this figure, it shows the Lynch syndrome. Many organs are suspected by the mutations in the Lynch syndrome. We have also here, we have a sort of translocations in the Burkitt's lymphoma between the chromosome number 8 and number 14. We have the Philadelphia gene. We have the BRCA1 and the BRCA2 genes. As I told you, they are present in the long arm of the Q21 chromosome. And this chromosome is formed of 24 exons. So if I want to make a correct genetic consultation, I have to know all the mutations all over the gene. These are the 24 exons, so I have to make the mutations all over the gene. It's not a matter of saying here there is a certain mutation, this lady is suspected to have breast cancer. This is not correct, because I have to make the consultation of the lady who is sick with breast cancer with her relatives, like her daughters or her sister. If both tissues are not present, this is not a sort of a correct genetic consultation, and thus will not be a hereditary cancer. The same applies for the BRCA2 gene, which is present in the long arm of the chromosome number 13, and it is formed of 27 exons. And again, I know some mutations are effective and others are not effective. And in Egypt, we don't know which mutation exactly is the cause of the breast cancer. Some races, like the Jews, because they marry each other from the same family, they marry each other, they know the hot spots for mutations that are suspected for breast cancer. Who are the people that have to make genetic consultation? Not everybody is liable for making this consultation, but if somebody has ovarian, fallopian, or primary peritoneal cancer, this is suspected, or this is important to make genetic consultation. Those who have breast cancer at an age younger than 50, those who have triple negative breast cancer, and this triple negative means that they are estrogen, progesterone receptor negative, and HER2, which is a growth factor, they are also negative. Those who have male breast cancer in their families, those who have pancreatic cancer, those who have prostatic cancer lower than the age of 55, younger than the age of 55, those who have Lynch syndrome or Codon or Lee-Froman syndrome. What is with and what is against genetic testing? Of course, the advantage of genetic testing is that they improve the risk assessment, they manage the increased risk. It relieves depressive symptoms. If I didn't have the mutation, this makes me relieved. Screening additional members from the family, influencing lifestyle and reproductive decision, management of the disease member with BRCA mutation or with any genetic mutation. What are the disadvantages? Its non-informative results might be deceiving. I might have some non-informative. I said we don't know what are the hotspots among the Egyptians. The sense of guilt may affect the parents. The sense of reassurance for those who are negative. I might be very relieved that I don't have this mutation. But in reality, the sporadic cancer is more than the hereditary cancer. It does not assure the development of cancer. And again, I will go to the molecular diagnosis, like the translocation in the chronic myelogenous leukemia, forming a BCR-ABL translocation on the gene, on the chromosomes, and this is known as the Philadelphia gene, and it is very informative for diagnosing this disease. Again, I have some markers that I have to search for in the breast cancer, like the HER2, and if I found that the patient is HER2 positive, her treatment will be completely different from the patient that is HER2 negative. If I have NMIC in neuroblastoma, this also directs the physician to use the correct treatment. And the same goes for the mutation in the P53, which is mutated in about 50% of the tumors, and it is a tumor suppressor gene. So what about the treatment? In the treatment, again, I will continue like the previous slide, that some markers indicate which treatment this patient must get, like the drugs which are interfering with the angiogenesis, like the endostatin and the angiostatin. So if I found the angiogenic factors high in the tumor of the patient, so he must be treated with a drug that faces the phenomena of the angiogenesis. Drug targeted against the oncogene PML, rare alpha oncogene in acute promyelotic leukemia, it's also very important. Drug targeted against the RES protein and drugs targeted against the CERB or the HER2. It is the same name, CERB, NEO, or HER2, the same name for the growth factors. Drugs against the BCR and ABL protein kinase, like the STI-57-1. This is a targeted drug against the disease. This is the drug which inhibits the HER2, and as you see, it closes the growth factor receptors on the membranes of the cell and causes the shrinkage of the tumor when the patient is treated with such a drug. I will move to the epigenetic changes, and epigenetic changes mean that the DNA sequence is the same, no mutation on the DNA, and the DNA is intact, but the word epigenetics means around or over. So some sort of mutations or some sort of changes in the DNA template occur and stops its replication or its transcription, like methylation of some islands present on the DNA and or ethylation of the chromatid spindles. You know that the DNA is surrounded by the chromatid and the histones. The histones, And there are a variety of compounds that are considered as epigenetic carcinogens, but they do not show mutagen activity. And these compounds are like diet, exposure to sunlight, exposure to some viruses, smoking, drug abuse, and even the financial status might cause epigenetic changes, or the social status. We find that if somebody was in Egypt and then he traveled to go to migrate in another country, some features changes in him. These features are caused by the epigenetic changes and not due to the DNA changes. That's why I can find in a certain disease that the DNA is intact, but the protein is not formed, because the DNA was not translated to the protein because it was inhibited by the epigenetic changes. As you see here, this is the epigenetic changes present either in the DNA itself or in the histones of the DNA, the histones surrounding the DNA. Some malignancies are formed due to epigenetic changes, genetic changes, and risk factors, risk growth factors like the oral cancer. We found that some alteration in the cell caused by the first mutation followed by the epigenetic changes and then followed by other chemicals causes the formation and the development of the oral cancer. This might lead us to forming targeted therapy against epigenetic changes and hitting the enzymes that are the cause of epigenetic changes like the HAT and the HDAC. We hit them with certain materials, and this is called the targeted therapy. We also have another molecular change which is the proteomic changes in the cancer. So what does we mean by proteomic? As I said before, that the DNA is transcribed into RNA, and the RNA is translated into proteomics, into protein, and we don't know all the proteins that we have in our body. And so the study of proteomics is to discover the proteins that we didn't study it yet, we don't know it yet. How can we know these different proteins that we have in our body? And what is the objective of studying the proteomics? It is to detect the different proteins expressed by the tissue, cell culture, or organism using the two-dimensional gel electrophoresis. We store this information in a database, like what we know as the data bank for the genome. We compare expression profiles between a healthy cell versus a diseased cell. The data comparison can then be used for diagnosing and drug design. How can we discover these new proteins? Of course, all of you know the one gel electrophoresis, one-dimensional gel electrophoresis. We separate the proteins in the serum or in the tissue in one direction, but in the proteomics we are using a two-dimensional gel electrophoresis. What is a two-dimensional gel electrophoresis? We first put the protein we have here in one of the wells or in the wells. We have a gel here which it's called the polyacrylamide gel, and it provides a matrix for the proteins to be separated. After separation, I have this skin. I can take this part and then inverse it and put it in another type of gel, which is the SDS gel. It will be further separated in the other direction. And finally, I will have a gel having this appearance with many types of proteins, which I knew few of them. I don't know all of them. So I will use two tissues, one from a patient and one from normal individual, and I will have 2 sheets like that. For example, I found that this protein is present in the diseased person and not present in the normal person, so this protein is the cause of cancer. And I have to attack this type of protein. How can I know this type of protein if it is very new for me? I have to determine the structure of this protein using polypeptide sequencing or the 3-dimensional X-ray crystallography and nuclear magnetic resonance and the mass spectrogram. All this will give me an idea about the groups present in this protein and the structure of this protein. I will take this protein and I will concentrate on it to separate the protein and to attack it and to imagine the active site of this protein, to inhibit the active site of this protein, and this is how I can form a new drug, or I can use it as a tumor marker for this disease. Another way to detect different proteins present in the tissue is to extract the protein from a diseased tissue and the protein from a control tissue, and they use the protein microarray. The protein microarray has a chip, and this chip, I don't know it is clear here or not, but it is formed of many probes that can attach the protein. And if this protein is present, again, if the protein, I found the protein which is present in the diseased person, this protein is my interest to form a drug against this protein or to use it as a tumor marker for a certain disease. This is a structure of protein that I have found, and I imagine that this is the active site. I imagine hundreds of compounds that can attack this active site of the protein. If I attack this active site with one of these compounds that I have imagined and I have synthesized, I will promote to the preclinical studies, which is on experimental animals, followed by the clinical trials on the human being, and following the ethics of development of a drug. To sum up, I have endogenous agents which causes cancer and exogenous agents which I have it in my body. I have inherited from from one of my parents, and this will cause DNA damage. This DNA can be intact. All the nucleotides are intact, but I might have further mutations like epigenetic mutation alterations, and this is caused by many other agents than those who attack the DNA itself and cause mutations. This DNA repair deficiency causes large increase in unpaired DNA damage, and this will cause large increase in somatic mutation and epigenetic alteration, and will cause alteration causing a disease, causing the progression of the cancer. Thank you for watching my presentation.